Drosophila larvae bearing a neuron-specific CD8 GFP fusion reporter transgene are used to isolate DA neurons. The larvae are dissected by inverting their cuticle inside out, exposing the peripheral nervous system. The inverted larvae are subjected to enzymatic dissociation, followed by mechanical dissociation via vortexing, trituration, and downsing. Cells are then filtered to obtain a single-cell suspension containing primarily epithelia, muscle, and neurons. Antibody-coated magnetic beads are added to the cell suspension, where they bind neurons expressing the CD8-tagged GFP fusion protein. Finally, the magnetic bead-coated neurons are isolated by placing the solution in a strong magnetic field, yielding highly purified neuron populations.
Hi, I'm Dan Cox in the Department of Molecular and Microbiology in the Krasnow Institute for Advanced Study at George Mason University.
Hi, I'm Ishwar Iyer from the laboratory of Dr. Dan Cox.
Today we will show you a procedure for the isolation and purification of Drosophila peripheral neurons by magnetic bead sorting.
We use this procedure in our laboratory to study the molecular mechanisms regulating class-specific dendrite morphogenesis. So let's get started.
To begin this procedure, pick 30 to 50 age-matched third instar larvae and place them in a 1.6 milliliter microfuge tube containing 1 to 1.2 milliliters of 1x PBS. Close the tube and vortex it at the maximum setting 3 times for 1 second each. Using a fire-polished Pasteur pipette, discard the supernatant completely, add 1 milliliter of PBS, and repeat the wash and vortex steps 3 times until the supernatant is visibly clear of any food particles and debris. Briefly repeat the wash with 1 milliliter of 70% ethanol, then wash the larvae twice with 1 milliliter of double distilled water, and finally repeat the wash once again with 1 milliliter of RNase away, and 3 times in 1 milliliter of double distilled water for complete removal of the RNase away. The larvae are now ready for dissection. For dissection, place 10 to 12 larvae on the center of a Silgard-coated 35 mm Petri plate. Position them slightly away from each other and discard any larvae that do not seem to be at the appropriate developmental stage. Next, use a pair of fine dissection scissors to cut open the anterior tip of the larvae. Using a pair of dull Dumont No. 5 forceps, invert the larvae inside out. Start by inserting one forcep inside the larval cuticle all the way to the posterior end. Pinch the tips of the forceps together to grab the posterior end of the cuticle. Pressing the cuticle down on the Silgard surface makes it easier. Using the second pair of forceps, push the larval cuticle inside out. Make sure the larvae are completely inverted so that all the soft tissues are exposed to the solution for easy dissociation. Practice this a few times before attempting to dissociate the cells. After dissecting 3 to 4 larvae, transfer them immediately to fresh, ice-cold PBS in a microfuge tube. Repeat until the required 30 to 40 larvae are collected before proceeding to remove nonspecific cells. Keep in mind that 10 to 20% loss during dissection and dissociation is expected. Now that all the larvae are dissected, clear loosely adherent nonspecific tissues, such as fat bodies and central nervous system cells, by replacing the supernatant of the tube containing the inverted larval cuticles with 700 to 800 microliters of fresh, ice-cold PBS. Lastly, pulse vortex the microfuge tube 5 times for 3 seconds per pulse at full speed, then discard the supernatant and replace it with approximately 700 to 800 microliters of fresh, ice-cold PBS. Repeat this step 3 times, and finally resuspend the larval cuticles in 400 microliters of fresh, ice-cold PBS before moving on to dissociate the cells. To isolate the dendritic arborization, or DA, larval neurons, use a combination of enzymatic and mechanical dissociation. Beware of overdissociation, which may cause the loss of the cell surface marker, leading to poor cell yield and low cell viability. Start the dissociation by adding 1.5 microliters of 1x liberase splenzyme 3 at 28 Wunsch units per vial to the larval cuticles suspended in 400 microliters of PBS. Vortex the solution 2 to 3 times for 1 second each at maximum setting, to remove the loosely adherent cells from the cuticles into solution. Then incubate the solution at room temperature for 5 minutes. Since incubation time greatly affects the final cell sorting efficiency, do not exceed 15 minutes. At the end of the incubation, pulse vortex the tube 20 to 30 times at full speed for 2 seconds per pulse. This should release the muscle and other non-specific tissue into solution. Inspect the sample periodically under a fluorescence-enabled stereomicroscope to determine if the peripheral neurons are still attached to the cuticle. Inspect a small sample from the solution by observing the microfuge tube directly under a fluorescence-enabled stereomicroscope to determine the level of dissociation. Continue to do this at each step of the dissociation. Stop vortexing before the peripheral neurons appear to dissociate from the cuticle into the solution. Briefly wash the larval cuticles with 2 to 3 exchanges of 1 milliliter each of fresh ice-cold PBS, and finally resuspend them in 500 microliters of fresh ice-cold PBS containing 1% BSA. To continue with the mechanical dissociation of the cells, pre-coat a 2 milliliter Conti's tissue grinder and large clearance pestle with a 1% BSA in PBS solution, and after a brief rinse, discard the BSA solution. This will prevent the larval cuticles from sticking to the glass surfaces. Subsequently, pre-cool the tissue grinder and pestle by placing them on ice for a few minutes in order to prevent cell damage or lysis. Then use a fire-polished Pasteur pipette to transfer the cuticles to the tissue grinder. Now douse the tissue with slow and steady strokes, avoiding frothing, for approximately 20 to 30 strokes. Make sure to douse slowly and steadily, otherwise the cells may lyse. To assess the cell dissociation levels, wipe the wall of the tissue grinder with a clean Kim-wipe tissue and inspect under a fluorescent stereomicroscope. The neurons should have detached from the cuticle and can be seen in the solution. A good indication of cell dissociation is the absence of neurons from the larval cuticle. However, if one still observes neurons attached to the cuticle or observes incompletely dissociated cells, douse further until the cells achieve a single cell suspension. Continue the mechanical dissociation using a fire-polished Pasteur pipette narrowed to approximately 50% of the standard tip diameter. Triturate the solution 5 times, followed by 10 times with a fire-polished Pasteur pipette narrowed to approximately 25% of the standard tip diameter. Note that forceful trituration may damage the cells. Monitor the cells between steps and adjust the procedure accordingly. Finish by filtering the solution through a 30 micrometer cell filter and collect the cell filtrate in a 1.6 milliliter microfuge tube. The resulting solution should consist of a single cell suspension and is now ready for magnetic cell sorting. Magnetic beads coated with an anti-CD8 antibody were prepared as described in the accompanying written protocol, and the DA neurons express a CD8 GFP fusion protein targeted by the antibody. Start the cell sorting by adding 15 microliters of antibody-coated magnetic beads to 500 microliters of cell suspension. Incubate the cells with antibody-coated magnetic beads for 30 to 60 minutes on ice with occasional hand mixing. Incubation at a higher temperature or longer time may result in nonspecific antibody binding. Next, place the microfuge tube in a strong magnetic field for 2 minutes. All positively selected cells along with unbound beads will adhere to the side of the tube. Slowly aspirate the supernatant without disturbing the cell pellet. Wash the cells 3 to 4 times in fresh ice-cold PBS. This step removes any remaining nonspecific cells. Finally, resuspend the cells in 30 microliters of fresh ice-cold PBS. To estimate the purity and yield of cells, pipette 5 microliters of the cell suspension onto the polished surface of a hemocytometer and count all the visible fluorescent cells under a fluorescent stereomicroscope. Also check the amount of non-fluorescent cells and any signs of impurities. Typically, the sample will be highly enriched for fluorescent cells. Finally, use the PicoPure RNA isolation kit to isolate RNA from the cells bound to the beads. Add the extraction buffer to the sorted cells, but remove the beads using a magnet prior to the column purification step. Use 1 microliter of the extracted RNA to assess total RNA quality on an Agilent Bioanalyzer 2100. RNA purified from the isolated DA neurons is of excellent quality, as indicated by the presence of sharp 5.8S, 18S, and 28S ribosomal RNA peaks when analyzed on an Agilent 2100 bioanalyzer. Beginning with 30 to 40 third instar larvae, we were capable of isolating, on average, 300 to 500 class 4 DA neurons using the PPK Gal4 driver, and 1500 to 2000 DA neurons, classes 1, 2, 3, and 4, using a pan-DA neuron-specific Gal4 driver. Quantitative RT-PCR with LAV and FUCH, which are neuronal-specific markers, revealed significant fold enrichment of both, indicating a highly specific enrichment for DA neurons using the protocol we demonstrated, as compared to flow-through. Finally, the isolated RNA from both pan-DA neurons and class 4 DA neurons was used to perform transcriptional expression profiling on Agilent Drosophila melanogaster whole genome oligo microarrays, identifying many factors implicated in DA neuron morphogenesis.
We have just shown you how to isolate and purify Drosophila peripheral neurons via magnetic bead sorting from the third instar stage of larval development.
When doing this procedure, it is important to balance the enzymatic and mechanical dissociation steps.
Excess enzymatic dissociation may lead to the degradation of CD8 cell surface markers, leading to inefficient or failed cell sorting.
It is also important to maintain RNase-free conditions throughout the procedure in order to preserve the integrity of the purified RNA from the isolated neurons.
So that's it. Thanks for watching and good luck with your experiments.